All right, here we're going to talk about proteins today and see what's going on with this next macromolecule. Proteins basically are the workhorse within the actual cell. The code for proteins is in nucleic acids, and the DNA, which is the nucleic acid, tells the cell which proteins to make. And depending on which proteins are made, different things are done. And if you look at this long list of things that proteins do, they pretty much do everything inside your body. They store things, they transport things, enzymes catalyze reactions, hormones are signals that are sent throughout your body, they regulate things. Basically, they kind of make your body work. So these ones are kind of, I mean, they're all important, but this one plays a very special role when we talk about the things that all the macromolecules can do. The monomer for protein, hopefully you remember that, is an amino acid, and that's going to get put together to create the protein. When we look at an amino acid, there's a couple things that we're always going to see. First is the alpha carbon. The alpha carbon is the thing that sort of holds I don't want to say it holds everything together, but sort of the central point when we are looking at proteins. Next to the alpha carbon, you're going to have your amino group on one side, and you're going to have your carboxyl group on the other side. Notice that the H has been left off, so it's C double bond O with a negative O because the H was donated as part of an acidic portion of this. Now, this is the part that makes all the difference. There's 20 different side chains for all the amino acids in your body. This top part right here, the amine group, the extra hydrogen, the carboxyl group, and the alpha carbon, that's always going to be the same in every amino acid. This side chain is going to determine how the protein actually folds on itself, which we'll talk about proteins folding in a second here. Now, when we look at this, this is a list of all the different proteins inside our cells, and you can see the different side chains involved here. With arginine, histidine, lysine have positive charges, and aspartic acid, glutamic acid both have negative charges. When we look at these are amino acids that are polar but uncharged side chains. So these ones are going to be polar. So hopefully when you hear the word polar, you think, hey, that's going to interact with water, or it's going to be hydrophilic. Same thing right there. Down here are the non-polar or hydrophobic ones. Those ones are going to not interact with water quite as well. And these ones are special cases. Depending on where you'd find these ones, they could do different kinds of things. Now, when you look at most of these names, which suffix do you see on the end of them? I see INE, 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 a lot of INEs. So if you see here a name, here's a word that has INE at the end, you gotta start thinking protein, think about the properties of protein. Again, that's that word association that I was telling you before that you need to sort of focus on in this course. When we talk about amino acids, I don't really care about oligopeptides, polypeptides, but one thing you do need to know about is the peptide bond. Peptide bond is extremely important. This is something that we only find in proteins. So when we're talking about associating certain words together, you hear the word peptide, you automatically need to think protein. And just sort of understand that those two words are synonymous. When you see the word polypeptide right here, that just means that there's many peptide bonds. And look at this, we got water coming out. Hopefully you're thinking of what kind of reaction that is. That is a condensation reaction, absolutely. And we look down, there's that peptide bond between the carboxyl group that was in one amino acid and that amino group that was in the other amino acid. So that's how the peptide bonds get linked together. We'll talk all about protein synthesis. That happens in like chapter 9 or so, somewhere around there. That all happens at the ribosome. That's where the proteins get made. But yeah, this is the big deal because these are the things that do everything inside your cell. The structure of protein is very important. Proteins are a 3D shape, and these can be ridiculously long chains. The largest protein in your body is something called titin. Titin is found inside muscle cells. It's inside something called sarcomere, and it goes from the length of the entire sarcomere. And that can be about 40,000 amino acids long chained together. So, I mean, this thing can get pretty big. If you have something that's that long, your body's natural thing, or the thing your cells naturally do, is they bunch it up and sort of fold everything onto themselves. And when we talk about structure of proteins, there are 4 basic structures to the proteins, and they all sort of have different features, and they all determine how the protein actually folds on itself and how it's used, because the folds of the protein expose certain bonding sites with that protein, which again, AP Bio always goes back to chemistry a little bit. The way those bonding sites are opened up determines what that protein's actually going to do inside the cell. When we look here, primary structure is all covalent bonds. Everything else has to do with hydrophobic, hydrophilic interactions, hydrogen bonds, ionic interactions. All of those things help them together. So let's look at some pictures of all of these, and then we'll talk a little bit more about them in a sec. When we look at primary structure, we see nothing but covalent bonds. Covalent bonds are those intramolecular bonds that hold the atoms together and start making the chain. You can see highlighted in red here are all the different peptide bonds. The peptide bonds hold the individual amino acids. The sequence of amino acids, again, is determined by the sequence of DNA that you have inside the nucleus. That sequence goes out to the ribosome through mRNA and gets translated into the actual protein. So one thing you need to know about primary structure, all covalent bonds, and this is sort of the base for the entire protein, which could get ridiculously long. Secondary structure can make one of two shapes. It can either make an alpha helix, which we see up on the top left, or it can make a beta pleated sheet, which we're going to see on the bottom right. Now, holding these alpha helix and beta pleated sheets together are going to be hydrogen bonds, which are interactions between the different R groups in the protein chain. We remember the amino acid has your alpha carbon in the middle, you have an amine group on one side, you have a carboxyl group on the other side of that alpha carbon, and the amine group and carboxyl group of neighboring amino acids is where the peptide bond is created. Then you have that R group, or the side chain, and that side chain is going to determine how it folds. Depending on the order of the amino acids, different side chains may be more attracted or less attracted to other parts of the protein chain. The polar ones are probably going to interact with each other. The non-polar ones are probably going to interact with each other. The charged ones, the positive and negative, will attract each other, and the positive would repel each other. So that's where that importance of understanding what the side chain is and being able to identify that kind of thing is important. Now, I'm never going to ask you to fold a protein. What you need to know is how the protein folds, though, but I'm never going to ask you to, if I show you a sequence of amino acids, ask you to tell me whether it's going to make an alpha helix or a beta pleated sheet. That would be crazy talk. We're not doing that in here. One thing you should know, though, is that you don't only have to have one alpha helix or one beta pleated sheet as part of secondary structure. There could be 50 of these and 100 of these. It could go back and forth. You could have just alpha helixes. You could just have beta pleated sheets. It just depends on the sequence that comes from the DNA. Tertiary structure is when we have this nice long chain of protein and we sort of see, I don't know if this ends a start or this ends a start, doesn't really matter. It doesn't really matter where primary structure is. We can see a beta pleated sheet as part of secondary. We see alpha helix over here, and different parts of the alpha helix sort of attract themselves to different parts of the chain of proteins. Same thing with the beta pleated sheet. We see hydrogen bonding right here, holding this end of the chain together. And tertiary structure is held together by a few different forces, which we'll get to in a second. Quaternary structures, and we have different individual subunits that are being held together. So this is almost like one individual protein, another one, and a fourth one, and they're all held together to act as one working protein. The classic quaternary structure that we see in AP Bio is going to be hemoglobin, which is a protein inside red blood cells. Hemoglobin's job is to attract oxygen and carbon dioxide molecules to transport them throughout the body. And that's just the classic quaternary structure. So if you ever hear this term, start sort of thinking hemoglobin, or if you hear hemoglobin, start thinking about this quaternary structure. Now, back to secondary structure a little bit. Hydrogen bonds between the amine groups and the carboxyl groups. That's what we're going to see right here. These red dotted lines all represent hydrogen bonds right there. Same thing in this picture on the right-hand side. We can see all the red dotted lines are hydrogen bonds that hold that chain of amino acids together in the individual shape. With the tertiary structure, I was talking about the different things that can cause the protein to fold on itself. Well, here's kind of a list of 4 of them. You can have covalent disulfide bridges. Now, you're only going to see disulfide bridges in amino acid side chains that have sulfur in them. means two. Sulfide is referring to sulfur. So if you have two of them that have sulfur in them, then they could create a disulfide bridge between them. Hydrophobic side chains are going to fold towards other hydrophobic side chains, and they're going to go usually towards the middle of the protein. Van der Waals are specific interactions that also work on the hydrophobic R groups, and that helps oils sort of make little dots inside water. That's considered van der Waal interactions if you ever pour drops of oil inside a glass of water. And you have other ionic interactions that are going to create charge chains and salt bridges that help fold that protein. Again, these structures can get really long, and the most efficient way to store them is by folding them into scrunched up shapes. Again, you hear quaternary structure, start thinking of hemoglobin. And that's when you have those 4 individual subunits that sort of latch together and act as one individual protein. Our last slide talks about what happens when the protein unfolds. When a protein unfolds, that is a process that's called denaturing. So when proteins denature, basically the secondary and tertiary structure sort of fail. The primary structure is always going to hold tight because it's got those covalent bonds, but the secondary tertiary structure that's held together by ionic interactions and hydrophobic and hydrophilic things and the salt bridges and the disulfide bridges, that can possibly denature. Two of the big things that denature stuff are temperature and changes in pH. When we talk about enzymes a little bit, that's a big thing that can affect how an enzyme works, and we'll talk about an enzyme being folded. That's another lecture though. You can also do it by submerging proteins in really high concentrations of either just polar or nonpolar substances that can cause the interactions to break down also. When that happens and the protein loses shape, it doesn't do what it's supposed to do. You might be like looking at this picture, getting a little hungry, being like, eggs look good, but what do they have to do with denaturing proteins? Well, the whites of egg is a protein. It's called albumin. When albumin gets heated up, change in temperature, then that protein denatures. And when egg whites denature, they turn white. And that's how you get fried eggs. And then we eat them, and that's kind of fantastic. So that's all I got for the protein background itself. But I do have another lecture coming up on enzymes that I'll get to you guys tomorrow. Hopefully you're having a good afternoon. I'll talk to you soon.